Welcome back to one more video on beta-oxidation of fatty acids part 2. In this video we are going to discuss the actual mechanism of the beta-oxidation. But before you watch this, please make sure to watch the previous part of transportation of the fatty acids, and the link is given in the description box below. Okay. In this second video of the beta-oxidation, we will see that how a 16-carbon fatty acid is broken down into its simple forms. In the previous video, we have seen that the palmitic acid is 16-carbon fatty acid, saturated fatty acid without any double bond, has been brought to target cell by the blood circulation and being converting into palmitoyl-CoA and then brought to the mitochondrial matrix by the carnitine transport system. Now this 16-carbon palmitoyl-CoA will undergo beta oxidation in the mitochondrial matrix and then be converted into a 14-carbon myristoyl-CoA and a 2-carbon acetyl-CoA. This reaction is catalyzed by 4 enzymes, and again this 14-carbon myristoyl-CoA will undergo again beta oxidation and forms a 12-carbon lauryl-CoA and a 2-carbon acetyl-CoA. Again it is cleaved into a 10-carbon compound and 10-carbon compound into a 8-carbon compound and 8-carbon compound into a 6-carbon compound and 6-carbon compound into a 4-carbon compound in each cleavage, yielding one molecule of 2-carbon compound acetyl-CoA. Now the beta oxidation will undergo 7 times, and the 7th beta oxidation will cleave this 4-carbon compound into 2 molecules of 2-carbon compound acetyl-CoA. So one molecule of the fatty acid will undergo 7 times beta oxidation in each cleavage, yielding one molecule of 2-carbon acetyl-CoA. So orally, the 16-carbon compound palmitic acid is converted into 8 molecules of 2-carbon acetyl-CoA by undergoing 7 times beta oxidation, in each cycle removing 2 carbons of the fatty acid chain. So the 16-carbon palmitoyl-CoA will be converted into 14-carbon compound and then 12-carbon compound and then into 10-carbon compound and then into 8-carbon compound and 6-carbon compound and 4-carbon compound, yielding acetyl-CoA in each step. Let us see the actual mechanism of the beta oxidation step by step. We have the 16-carbon palmitoyl-CoA. It is a saturated fatty acid. It having the 16-carbon chain here, which is an acylated form attached to the coenzyme A, having the alpha and the beta carbon here. The first step in the beta oxidation is the dehydrogenation. In this step, we are going to remove the 2 hydrogens from the beta and the alpha carbon, and we are going to give it to the FAD+, converting it into FADH2. This reaction is catalyzed by the acyl-CoA dehydrogenase. After removing the hydrogens, now there is a double bond between the alpha and the beta carbons. This structure is called as a trans-enol-CoA, as the hydrogens are in the trans position. The next step is hydration. In this step, we are going to add water molecule to the above compound, which is catalyzed by the enol-CoA hydratase. We can cleave the double bond by adding the water molecule. That's why the reaction is called hydrolysis. After adding the water molecule, the double bond between the alpha and the beta carbon is cleaved, and it is forming a structure called as a hydroxyacyl-CoA. The next step is also the dehydrogenation, but in this dehydrogenation, we are going to remove hydrogens only from the beta carbon, giving that hydrogens to NAD+, yielding NADH. Now, after removing hydrogens from the beta carbon, what happens is there will be a double bond formed between the carbon and the oxygen. And by this, we got a symmetrical compound, and this is called keto-enol-CoA. This completely symmetrical compound. This reaction is catalyzed by hydroxyacyl-CoA dehydrogenase. Now this last reaction is the actual cleavage. The enzyme thiolase cleaves the above compound at beta position into a 14-carbon compound myristoyl-CoA and also a 2-carbon compound acetyl-CoA. This reaction is called as the thiolysis because the thiolase enzyme cleaves this into two forms. The 14-carbon compound can re-enter the reaction and can perform the beta oxidation again. Now just look at the mechanism carefully. You can see that the first three steps are to just prepare the palmitoyl-CoA for the cleavage. It is actually the fourth step which actually cleaves the palmitoyl-CoA into 14-carbon compound myrstol-CoA and 2-carbon compound acetyl-CoA. Now when you look at an overview of the beta oxidation, we got the 16-carbon compound palmitoyl-CoA has been converted into a 14-carbon compound myrstol-CoA and also a 2-carbon compound acetyl-CoA by undergoing beta oxidation, which is catalyzed by 4 enzymes. Now this 14-carbon compound will re-enter again into beta oxidation to convert into 12-carbon compound and acetyl-CoA. And again this 12-carbon compound will re-enter the beta oxidation cycle to convert to 10-carbon compound and 10-carbon into 8-carbon compound and 8-carbon compound into 6-carbon compound and 6-carbon compound into the 4-carbon compound. In each step, cleaving the molecule into 2 molecules, 1 molecule of the carbon main chain and 1 molecule of the acetyl-CoA. So after the 7 cycles of the beta oxidation, we now got 8 acetyl-CoA molecules, 7 molecules of NADH and 7 molecules of FADH2. The acetyl-CoA will be given to the Krebs cycle for yielding ATP, and the 7 NADH2 will be given to the electron transport system, and same for the FADH2, which will happen in the mitochondrial matrix. Let us see the energy yield of the beta oxidation. After the 7 cycles of the beta oxidation of fats, we now get the 8 molecules of the acetyl-CoA, the 7 molecules of the NADH and 7 molecules of FADH2. But the question is, how much amount of ATP this all molecules yield? Before you think that, how much amount of ATP that one acetyl-CoA yield? To answer that, how much amount of ATP one acetyl-CoA yield, remember, when one acetyl-CoA is taken by the Krebs cycle, 1 molecule of acetyl-CoA can yield 3 NADH, 1 FADH2 and 1 ATP. To know how many ATP molecules they all yield, remember, the ATP synthase in the inner mitochondrial membrane can pump 4 protons from the periplasmic space to yield 1 ATP. So if that is the case, if 1 NADH supplies you approximately 10 protons, then 1 NADH can yield you approximately 2.5 ATP, 1 NADH2, and 1 FADH2 can yield you 1.5 ATP. So the 3 NADH above can yield you 7.5 ATP and 1 FADH2 can yield you the 1.5 ATP. So orally, 1 acetyl-CoA in the Krebs cycle, 1 acetyl-CoA can yield you 10 molecules of ATP. Then 8 molecules of acetyl-CoA can yield you 80 ATP and 7 molecules of NADH can yield you 17.5 ATP and 7 molecules of FADH2 can yield you 10 ATP. Orally we got 108 ATP, but we are minusing 2 because 2 are used for the palmitoyl-CoA activation. So orally 1 molecule of palmitic acid can give you 106 ATP. So finally, from the beta oxidation of 1 fatty acid, we have yielded 106 ATP. You know, this is thrice the energy harvested than 1 molecule of glucose. That's why the fats have the very high amount of energy. Let us see the advantages of storing the triglycerides as body fat. As we have previously discussed, triglyceride means 3 fatty acid chains attached to the 1 glycerol head. Mammals, such as the world's largest animal, the Balanoptera musculus, the blue whale, can store these triglycerides as body fat under the subcutaneous tissue in the form of blubber, which can give its insulation in the cold Antarctic waters. Humans also store the triglycerides as body fat in subcutaneous tissue and abdominal layer, which can vary in both the sexes, in males 21% and females 28%. The main advantage is that the carbon in the triglycerides is in highly reduced form, and and it is hydrophobic in nature. So the animals which store body fat in the form of triglycerides are so advantageous. That's all for the video. Please make sure to subscribe the channel Akhil's Biology Pandora and tell me in the comment section what I should do next. Thank you for watching.